This is the muscular system skeletal muscle contraction lecture. Here are your learning objectives. We're going to jump right in by starting to talk about the neuromuscular junction. So we haven't done the nervous system yet, but this is kind of a good introduction to thinking about how signals are passed through your body. So neurons that control effectors, okay, it's a word, a thing that has an effect. We've talked about effectors before. Such as skeletal muscle are called motor neurons. So you've got lots of neurons coming out of your brain and out of your spine that are controlling your movement, and those are motor neurons. Skeletal muscle, smooth muscle, whatever they're moving, whatever they're affecting, that's still going to be a motor neuron doing it. Each skeletal muscle is connected functionally, but not physically. It's a functional connected, meaning they affect each other, but they don't actually touch. So the skeletal muscle fiber is connected functionally to a motor neuron that extends from the brainal spinal cord at a space called a synapse. And I'm going to show you a picture of a synapse. Neurons in general communicate with other cells by releasing something called a neurotransmitter at a synapse. So they communicate with skeletal muscle, smooth muscle, glands, tissue, other neurons, anything else they want to communicate with. They use these chemicals called neurotransmitters. They release them at a synapse, and the chemicals have their effect on whatever cell they're going for. The site where the motor neuron axon, okay, so that's the end of the motor neuron, meets the muscle fiber is called the neuromuscular junction. A single motor neuron and all of the muscle fibers that it connects to form a motor unit. So one single motor neuron can connect functionally with lots of different muscle fibers, and that's a motor unit. A single motor neuron and all of the muscle fibers that it connects to. At the neuromuscular junction, which is here, okay, so neuromuscular junction, where the neuron meets the muscle, you have a motor end plate, which is the area of the muscle cell region that's specialized to form the neuromuscular junction. So this is the muscle cell, and you can see it has these folds kind of around the end of this motor neuron axon. So it's kind of folded and just specialized to meet these synapses. Lots of mitochondria around there, needs lots of energy, and the sarcolemma is folded. Here is a close-up of what one of these synapses looks like. So that's the end of the axon, this bulbous shape, and you can see these synaptic vesicles, okay, so there's an organelle, releasing neurotransmitters. In this case, the neurotransmitter is acetylcholine into the synapse. So the acetylcholine is released by exocytosis into the synapse. Then the acetylcholine will have its effect on this muscle cell. There are two videos here. I'm going to let you click on these links. I'm not going to put it in the recorded lecture, that are really helpful for understanding the events that occur at the neuromuscular junction and the excitation contraction coupling stage. I'm also going to talk about these in the lecture, but please do watch the videos. They're very helpful. So these are the events at the neuromuscular junction. The first thing that happens is you have a nerve impulse, which is an electrical signal that travels down the motor neuron axon and causes the release of acetylcholine by exocytosis into the synapse at the motor end plate. The acetylcholine binds to receptors on the muscle cell. So the receptors are proteins that are specialized to recognize acetylcholine and let it bind. When the acetylcholine binds to the receptors, this opens ligand-gated sodium ion channels. So that was a lot of words, okay? So a ligand is something that binds to a receptor, right? I said it was ligand-gated. So that means that when the chemical binds to the receptor, a gate opens. And in this case, the gate that opens is a sodium ion channel. Sodium ion channels, and then that makes the sarcolemma permeable to sodium ions. Okay, so when the receptor binds, sodium can enter the sarcolemma. Sodium can cross the sarcolemma and enter the muscle cell. So acetylcholine binds the receptors. That opens the sodium ion channels so the sodium can diffuse into the sarcolemma, into the sarcoplasm. That is, if you remember, a type of facilitated diffusion, an ion channel. So entry of the sodium into the sarcoplasm stimulates a muscle impulse. Okay, so we've had a nerve impulse, which is an electrical signal traveling down the nerve, and now we have a muscle impulse, which is an electrical signal, and you're like, electrical, what? But sodium is a charged particle, it's an ion. So we're changing the charge of the muscle, which stimulates a muscle impulse. All that sodium going into the muscle stimulates the muscle impulse. The muscle impulse travels through the muscle fiber on the T tubules. If you remember from the last lecture, the T tubules go inside all the muscle fibers, and they cause calcium to diffuse out of the sarcoplasmic reticulum. So think back to that triad arrangement of the cisternae of the sarcoplasmic reticulum next to the T tubules. When the impulse travels down them, they cause calcium to diffuse out of the sarcoplasmic reticulum. Calcium binds to troponin and causes a conformational change which exposes the binding sites on the actin molecule. So the actin, troponin, and tropomyosin complex, calcium binds to troponin, which pulls the tropomyosin off the actin binding sites and exposes them. The myosin heads can form cross bridges and bind to the actin, which starts the contraction. Okay, that is a lot of information, a lot of things going on, and it's just kind of a quick overview of the muscle contraction. You really need to break all of these pieces down, watch the videos, and when you do this in class, we'll really go through it one by one in great detail. So I want to talk about the sliding filament model of muscle contraction. So I just gave you the kind of quick, all the chemicals, the calcium, how everything is changing, but what actually happens when the actin and myosin bind? So here's your sarcomere, right? Z line, A band, the M line would be here. And you have the thick myosin filaments and the thin actin, troponin, tropomyosin filaments. When the muscle is relaxed, nothing is happening. When calcium binds to troponin and that pulls tropomyosin off of the actin binding sites, then the myosin heads, which you can see here, attach to the actin. Okay, so these little myosin heads just come on up and they bind to the actin. They actually just start pulling the actin filaments towards each other, and the muscle is contracting until the actin filaments go all the way in and the muscle is fully contracted, fully shortened. I want you to watch this video as well. It shows the cross-bridge cycle, which is the actin binding to the myosin bit by bit by bit, and you can see how the myosin heads pull the actin together to make the muscle shorten. Here's the image of the cross-bridge cycle, okay? Called the cross-bridge cycle because when the myosin binds to the actin, that is called forming a cross-bridge. So here's a relaxed muscle, okay? There's a thick myosin filament and the thin filament, which includes the tropomyosin, troponin, and actin. When you add calcium from the sarcoplasmic reticulum, the calcium binds to the troponin, you can see that here, which pulls the tropomyosin into the groove between the actin molecules, right? So that looks very different. This is the tropomyosin covering the binding sites. This is the tropomyosin releasing the binding sites. So these light pink sites on the actin are where the myosin heads can bind. When they're exposed, the myosin heads bind to the actin molecules. Okay? You have ATP here, which is ADP plus phosphate, making ATP. Okay? The cross bridge is formed. The phosphate is released from the ATP. Okay, so you can see there it's added on there. Here it's released, and that causes the myosin heads to pull the thin filaments. That's the power stroke. The ADP and the phosphate are both released, and then you add another ATP to break that cross bridge and reset the myosin. and start the cycle all over again. And I understand that this is really complicated and hard to pick up, but again, we'll go through it a few times, and that video is also very helpful. So the breakdown of the cross-bridge cycle in words: the ATP binds to myosin and breaks down, which is hydrolyzing to ADP and phosphate, putting the myosin head into a cocked position so it can attach to actin at the binding site. The ATP is released and the power stroke pulls the actin, causing greater overlap of thick and thin fibers and shortening the sarcomere. Then another ATP binds to the myosin and releases the first cross bridge, readying it to bind again. The cycle continues as long as ATP is present and acetylcholine is being released at the neuromuscular junction. So here you need ATP to bind, to form the cross bridge, to pull, and then to break the cross bridge as well and form a new one. For the muscle to relax, you have an enzyme called acetylcholinesterase. All right, so ACE enzyme breaks down acetylcholine. Calcium ions move back into the sarcoplasmic reticulum. The cross bridge linkages break. Tropomyosin, since the calcium is gone, rolls back into its groove, which prevents the myosin from binding to actin, and the muscle can relax. So this is the major events of muscle contraction and relaxation. They have 10 steps here, okay? I probably would think that there's a little bit more than that, but we will talk about them in class. And then, of course, relaxation as well. So you should be able to describe the events that take place during a complete contraction cycle, from initiation of the nerve impulse all the way through to relaxing the muscle. So just to talk a little bit about muscular responses. So muscles have a threshold stimulus. The threshold stimulus must be reached in order to stimulate an action potential, which spreads through the muscle fiber, releasing calcium and activating the cross bridge. So the nerve impulse in the motor neuron usually releases enough acetylcholine to reach the threshold stimulus and evoke a muscle impulse. Threshold stimulus is the minimum amount of stimulation you need to actually activate the cross bridge. A twitch is the contractile response of a single muscle fiber to muscle impulse. So one muscle fiber to one muscle impulse. And a twitch looks like this. So this isn't something that you ever see in real life in your body, but it's what you can measure. So this is a single muscle fiber. You have a latent period where nothing happens right after it's stimulated, and that's when kind of everything, calcium is being released, everything is getting ready to contract. You have a period of contraction and a period of relaxation. So this is one muscle cell contracting to one nerve impulse. Now when that happens a lot, you actually have a visible contraction. That happens by summation. So when individual twitches begin to combine without completely relaxing, it becomes a sustained contraction. So these are basically all of our contractions that we make, are combination of individual twitches. The force of the individual twitches combines by summation. And when there's not even a partial relaxation between twitches, the resulting forceful contraction is a tetanic contraction, and that is shown here. So this is a summation, right? Individual twitches here. Summation, so you have greater force resulting in contraction, and contraction without even a little bit of relaxation is a tetanic contraction, and you've probably heard of tetanus, right, or lockjaw. So the effects of tetanus are to cause a tetanic contraction where there is no relaxation. Lockjaw, right? Contracting. So the figure again shows a series of twitches, summation, and a tetanic contraction. In the body, most of your muscles are actually continuously partially contracted. That is what muscle tone is. So just allow you to stand still or just sit up straight. Your muscles are contracting slightly, and that is called muscle tone. And talk about muscle force. There are a few things that affect how forcefully your muscles can contract. And one is the baseline muscle fiber length, and that's what this is showing. So muscle fiber length when it's relaxed. If the muscle is overly shortened and relaxed, and you can think about this, if you bend your elbow and hold it, flex it, not all the way, but pretty close to all the way. And then if you contract it fully, you're not going to generate as much force because those muscle fibers are already shortened. Same thing if you stretch it all the way out and your muscle fibers are overly stretched, you're not going to generate as much force. If you have your muscle fibers at an optimal length, which is about where they should normally be for rest, then you can generate the most force in your contraction. The other two things that change how much force you can generate is the frequency of stimulation. So if the nerve is firing quicker and quicker and quicker, you're going to have a more forceful contraction. Also, how many fibers are stimulated during contraction? Are you stimulating just one fiber, or are you stimulating 500,000 fibers? More fibers is a bigger contraction, more force. And that has to do with recruitment. So if you remember what a motor unit is, it is the one nerve and all of the muscle fibers that it is attached to. So one motor neuron and all of the muscle fibers that it's attached to. This is a functional unit. So a nerve impulse in the motor unit will contract all of the fibers in that unit at the same time. Here's motor neuron for motor unit 1, and it's got a few points that it contracts to, a few different fibers, and this one has fibers that it attaches to too, okay? They separate them by color. So motor unit 2 has the red ones and motor unit 1 has the purple ones. So an impulse on this motor neuron will contract the purple fibers. An impulse on this motor neuron would contract the red fibers. But a muscle is composed of lots of different units and they all have different thresholds of stimulation. So, if only a few motor neurons are stimulated, only a few motor units will contract. If the stimulus intensifies, more motor units will contract, generating more force. And this is called recruitment. And to explain this, I like to tell a story, a very kind of funny story. But you can imagine this. So, you're holding a very flimsy plastic cup. Okay, if you squeeze it, it will spill. So you're cupping this cup in your hand and you're using only a few motor units, right? Someone hands you a heavy glass mug into the same hand. You put your plastic cup down, you pick up your heavy glass mug. You're using a lot more motor units to do the same action. You're having to recruit more motor units because you need more force to hold that heavy glass mug instead of that light little plastic cup. Okay, so that should make sense, right? You need more recruitment, you need more motor units for generating more force to hold the heavy glass mug. So on the day I got married, I was holding a heavy glass mug. And it was right after our ceremony, before we'd taken any professional pictures, and I took the heavy glass mug and I put it down, and a second later someone handed me a flimsy plastic cup with red wine in it. And so what did I do with that flimsy plastic cup? I squeezed it and spilled red wine all over my wedding dress before we'd even had pictures taken. So recruitment, it's important to temper how much contraction you need in a unit. And normally your body does that, but I didn't do it very well on that particular occasion. Anyway, types of contractions. Okay, so in an isotonic contraction, you change the length of your muscle. In a concentric isotonic contraction, you shorten it. In an eccentric isotonic contraction, your muscle lengthens, okay? So this muscle is contracting against this force, but the weight is moving down. In an isometric contraction, there is no change in length. So this person is just grabbing onto this bar and it is not moving, but that muscle is contracting to apply force against it. You've also probably heard of fast versus slow twitch muscles. So slow twitch muscles are red fibers and they have a slow contraction speed relatively. They're fatigue resistant and they have a really good blood supply. So people that have slow twitch muscles would be like long distance runners or long distance swimmers, people that have major endurance, do a lot of endurance exercise. Whereas fast twitch muscles contract rapidly, they're easily fatigued, and they have a poorer blood supply, making them white fibers. So these are the muscles that you want in a sprinter or a weightlifter. Fast twitch, rapid contraction, very forceful. So swimming and running, what type of fibers do they activate? Well, these would be the red fibers, okay, the slow twitch, and what physiological changes would take place with this kind of regular exercise? So if you're developing your slow twitch fibers, you're going to develop more mitochondria, so they're more fatigue resistant, and extensive capillary networks to constantly deliver oxygen to those muscles and increase their fatigue resistance. The muscle shape and size does not change very much. Whereas weightlifting, you're primarily activating white fibers. And what you're going to do with those is synthesize new filaments of actin and myosin, and that increases the diameter and changes the shape and size of the muscle. Another clinical thing is a disorder called Myasthenia gravis. The immune system produces antibodies to and attacks acetylcholine receptors at the neuromuscular junction. So it's an autoimmune disorder, okay? In this case, your body has decided that it doesn't want your acetylcholine receptors to help your muscles contract. So in this case, you get weak, flaccid-looking muscles. And to treat this, what you would actually give is an acetylcholinesterase inhibitor. So if you remember, acetylcholinesterase is the enzyme that breaks down acetylcholine. So if you inhibit that, then you're going to extend the action of acetylcholine for whatever receptors are remaining. Lastly, I also want to talk about rigor mortis. So rigor mortis is the effect on the body after death where the whole body just kind of freezes up into one position and can't be moved. The cause of that is when you run out of ATP. So you lose all your ATP, and ATP is required to form the cross bridge for muscle contraction, but it's also required to break the cross bridge. And so what you have is an effect where the cross bridges are all formed, the muscles are contracted, but the ATP is gone and they can't be broken. So you actually will stiffen up until the muscles themselves begin to degrade. All right, I said I was going to talk about visceral smooth muscle in this lecture, and so it is single unit smooth muscle. And its contraction is that it responds as a single unit. So when one fiber is stimulated, the impulse moves over the surface and stimulates others. This is different from skeletal muscle, where each fiber must be individually stimulated by a motor neuron. Here, one fiber gets stimulated and the impulse moves over the surface and stimulates others. It's also rhythmic. You have a pattern of spontaneous repeated contractions. So now, if you can kind of imagine this, you have rhythmic contractions and impulse moving over the surface like a wave. So these two features, the cell-to-cell stimulation and rhythmicity, are responsible for something called peristalsis. 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 It's pushing things through. And it's found, of course, in the digestive system, where you're pushing food through the tract. Smooth muscle contraction, it's like skeletal in that it's triggered by membrane impulses and the release of calcium ions and uses energy from ATP. It's unlike skeletal in that there's no troponin. Instead, calcium binds to calmodulin. Calcium comes from the extracellular fluid, not the sarcoplasmic reticulum. It can be stimulated by acetylcholine and norepinephrine, and can also be stimulated by hormones such as oxytocin during childbirth. Stretching can stimulate contractions, which is important for things like your bladder, right? So stretching of the bladder will stimulate the bladder to contract and try to void, which then you have a skeletal muscle which actually controls that. Slower to contract and relax, but can contract more forcefully for longer periods with the same amount of ATP, so it's more efficient. And you have the movement, which is called peristalsis. Okay, so that was, again, kind of a a quick run through of skeletal muscle contraction, which is an extremely complex process, but I wanted to give you this up front and then also go over it with you in class, but hopefully you've already had some exposure to it. So watch the videos, listen to the lecture, and I will see you next week.